Okay, so today's topic of discussion will be antiviral drugs for non-retroviral infections. Before we talk about the drugs about this particular pathology or antivirals, we first need to look at a little bit of microbiology of the viruses. So first let us discuss the basic components of a virus particle, or which we usually call it as a virion. Okay. So, as you already know, these are microorganisms that contain a core of genome of nucleic acids. So, it could be either RNA or DNA. So, they contain a nucleoproteins that could be your RNA, or these could be your DNA, anything, right? Now, surrounding this particular structure will be a protein shell, and this protein shell is important for the bacteria as it helps in various processes of it. So we would like to call that structure as a capsid layer. So this is circular, don't mind the drawing. So this particular structure that I'm drawing is your capsid layer that covers the genetic material. Now, most of the viruses, they have an outermost layer also, and that is made up of a layer of lipoproteins. And that lipoproteinaceous structure is referred to as an envelope. So on this envelope, you will find many glycoproteins also attached, right? Like you might have studied in your microbiology, they are referred to as hemagglutinin or neuraminidase glycoproteins. These are they look like spikes or small balls on them. So they are basically referred to as peplomeres. This is the capsid layer. Please mute your audios. And this is the genetic core. So since we are not studying microbiology right now, we'll leave this structure as it is, and the genome or the genetic core plus the capsid layer plus the envelope is referred to as virion. Right? So now next, the important thing that you have to see over here is the classification of viruses. How are the viruses classified? And if you have completed your virology in your microbiology, you might actually know this. But what I'm going to do is I'm going to briefly summarize what are the different types of viruses that you will be studying. Now they are classified depending upon what type of genetic material they have, and rightly so, they are divided into DNA viruses, right? And they are divided into the RNA viruses. Please mute your audios. Now, these DNA viruses, first up, we have your adenovirus. These particular adenovirus, if you remember, they cause usually your upper respiratory tract infections. Right? And then we have your hepadnavirus. The hep stands here for your hepatitis, mostly your hepatitis B. And third, you have got your herpes virus. Now, herpes virus is a very big family, and you might remember that you have your herpes simplex virus type 1, then you have got type 2 of it, and then we have got varicella zoster virus that causes chickenpox in children and shingles in adults. Then we have got cytomegalovirus that causes certain diseases. Mostly it causes infectious mononucleosis. And then lastly, we have got your Epstein-Barr virus. Cytomegalovirus, yeah. and your Epstein-Barr virus. So this is pretty much a very big family of herpes virus. And then we have got your papilloma virus. Papilloma virus usually cause spots in human beings. Then lastly, we have got pox viruses, as the name itself suggests. It causes smallpox, but this has been eradicated. And sixth, we have got your parvoviruses. Not that important, but if you want to know, they cause erythema infectiosum. Right. Now moving on to the RNA viruses. RNA viruses, we have got a pretty big list. And the first up is your picorna virus. Picorna virus is quite famous with you because many of the superstars advertise about it. They are the brand ambassadors. Polio. Then you have got orthomyxovirus. We'll be studying about the life cycle of this particular virus because they are influenza type virus, influenza A, B, and the rest. And then we have got paramyxovirus that causes mumps. Then fourth, we have got rhabdovirus, causes rabies. Then we have got arbovirus that causes your chikungunya. Just look at the spelling, it's not chikungunya, it's chikungunya. Then rotavirus. If you have been listening to your SPM classes, you might have heard about this virus. It causes gastroenteritis in children, right? And then you have got your retroviruses. This particular chapter is about non-retroviral infections. Retroviral infections will be dealt in the next class. So as you all know, this is all about the HIV. Then eighth, we have got Rna virus. Rna virus causes viral meningitis. And the most dreaded that is causing pandemic right now is your coronavirus. So this is the classification of virus. If you want to put a mnemonic to it, you can as well be my guest, prepare a mnemonic and remember these viruses that are classified into DNA and RNA. Right. Now, after having studied the type of viruses and the basic structure of viruses, let us go through the replicative cycle of a DNA virus. Right. So we'll be studying about the life history of this DNA virus as to how it looks like, how it enters the cell, and what is the life cycle of it, and how does it get released from the host cell, right? And what are the drugs that act. So let me take a fresh page and let's talk about the replicative cycle of a DNA virus. So you already know the structure or the basic structure of a virus particle, virion, is it contains an envelope, right? And it also contains papillomas, they are like spikes on the outermost surface. And then it contains what? It also contains your capsid layer. I'm going to just draw it as a single layer without the beads. And then inside you'll have the genetic core. So this is your double stranded or your genetic material, imagine. Right. Now, this virus, through its specific receptor sites, it goes and binds to specific locations on the host cell. Right. So let me draw a host cell for you. Now, this is the host cell. This is your body cell, right? Now, what the virus does is it keeps on searching specific areas on this particular host cell to make its entry. And when it has found its specific location or its the favorite site for it to enter, it goes and induces the process of endocytosis. What do you mean by endocytosis? The cell is taken, the virus cell is taken inside the host cell. That is your endocytosis. Right. So this is what I'm drawing is the process of endocytosis, and this is the second step. First step is the virus locating the specific cell surface on the host cell. Okay. After having done this, after the process of this is the first step, this is the second step. Now, after having done this process, only the genetic material of the virus is allowed to go inside. So this core, genetic core that contains the virus, that contains the genetic material, enters the cytoplasm. Now from the cytoplasm, this particular genetic core goes into the host cell nuclear site. Okay, so this is what happens. Now once inside, this particular genetic material, let me draw it over here, right, is acted upon by the host cell's RNA polymerase. Right. Now this host cell polymerase, which is called as RNA polymerase, converts it and causes the transcription and translation process and converts it into the viral mRNA. The genetic material of the virus is acted upon by the host RNA polymerase, and it gets converted into a viral mRNA. Now this viral mRNA is quite smart, and what it does is it utilizes the ribosomes, which is the 50S and the 30S subunit, right? It utilizes that and starts the transcription and translation process. And the first protein that it forms via this step is your regulatory protein. Now, why am I mentioning the first step? Why am I mentioning this protein in a stepwise? The reason is, after the formation of regulatory proteins, these regulatory proteins that are synthesized first, what they do is they initiate the transcription of early genes that are responsible for replication of viral DNA by viral DNA polymerase. So what happens is, this regulatory proteins, what they do is, if you look carefully over here, this is the viral DNA polymerase acting on the genetic core, and it helps in the formation of new strands of viral DNA. Now this viral DNA, how did it come into existence? It was because of the action of regulatory proteins. These regulatory proteins, they initiated the transcription of early genes, and that caused the replication of this viral DNA. That means they initiated this particular process. Now this viral DNA that you have got, this viral DNA, what it does is goes and forms another set of proteins which are important for the structure of the viruses, and those are called as structural proteins, right? And lastly, we have got one more proteins that are formed, and these are your viral enzymes. Enzymes are nothing but proteins, right? So through the process of transcription and translation of the viral mRNA, regulatory proteins are formed, and this regulatory proteins will initiate the formation of this viral DNA. Now this viral DNA helps in the formation of another set of proteins which are called as structural proteins, and lastly we have got your viral enzymes. Now, all these three proteins now result in the assembly of virions. Now we have got all the spare parts. Now it's time to put all of them together. And when you put all of them together, what happens? You get a virus particle which is called as virion, containing of an envelope, right, of made up of lipoproteins. And then we have got a capsid layer, and then we have got your gene core. So after having learned the replicative cycle of DNA virus, it is now time for us to put in certain drugs. So the first set of drugs that we have, they are called as DNA polymerase inhibitors. So let me write it over here. They are termed as DNA polymerase inhibitors. Now, in the DNA polymerase inhibitors, they are of three types. The first type is your purine analogs. The first type is your purine analogs. Now, what are the drugs that you will see in these purine analogs? The drugs are acyclovir. Then you have got valacyclovir. Then you have got ganciclovir. And then you have got valganciclovir, and there are a few more. So if you want to take a look at the extra drugs, you can feel free, be my guest, open up your textbook and see. So, but our point being, after having studied the drugs, you have to know where does this act in this particular diagram. So the purine analogs, they act at the level of the viral DNA polymerase. So this is where your number one drug acts. So when it acts over here, what it does is it inhibits the viral DNA polymerase action of the viral DNA polymerase activity. So when the viral DNA polymerase activity gets inhibited, what do you think will happen? The structural proteins that were bound to happen, that were bound to form due to the activity of this particular viral DNA will not form. And when the structural proteins are not formed, the basic structure of the virus particle is not formed. And when that is not formed, there's no point of them being released from the host cell. So that is how your purine analogs acyclovir act. Now the second type of drugs that we have over here are your, let me write it over here, are your pyrimidine analogs. So you have just got three drugs to remember over here, and the first is idoxuridine, second is trifluridine, and the third is telbivudine. I know these are tongue twisters, but take them as a challenge and learn them. It's fun. So since these are pyrimidine analogs, most usually they also act at the level of the viral DNA polymerase again. So it's one thing less that you have to remember is that purine analogs and pyrimidine analogs, they act at the same level. Now, talking about the non-nucleosides. Non-nucleosides, we have just got one drug, and that is your foscarnet. So we have a drug which is included in the group of non-nucleosides, and whenever I study about this drug, or take the name of this drug, I'm reminded of Skynet of Terminator. You know, it's Foscarnet and Skynet. They kind of sound similar. So that is how I remembered this name, and it's one of my favorite drugs. So what this Foscarnet does is it also acts at the level of the viral DNA polymerase. But the way it acts is it causes some disturbances in the phosphate nucleotides of the viral DNA polymerase. So if you want, you can go ahead and write number 3 again at the level of the viral DNA polymerase. So this is how the drugs that are related with the DNA viruses act. Right. So they act mostly at the level of the viral DNA polymerase. So we have got drugs that are included under DNA polymerase inhibitors, and we have got 3 types, and those are purine analogs, pyrimidine analogs, and non-nucleoside drugs, which include one drug, and that is foscarnet. So after having dealt with the replicative cycle of a DNA virus, let us talk about the replicative cycle of an RNA virus. Now, here in this case, what we need to find out, or what we need to do is, we have to go through the similar structure as to how we studied about the DNA virus life cycle. All right, let us proceed towards the replicative cycle of a RNA virus. So here, what I'm going to do is, I'm going to take the example of influenza, because that is most important virus among the RNA family. So let us draw a structure of the influenza virus. So as you all know, it has got an envelope, right? Now on this envelope, there are certain glycoproteins. They are referred to as peplomers, right? And we have got spikes on it, right? And these spikes are referred to as hemagglutinin. Now, depending upon the hemagglutinin and the neuraminidase enzyme, these are divided into subfamilies, if you have studied your virology. Right. Now, these spikes that you see, these are referred to as heme agglutinin. Now, the point of concern for us is these small green dots that I am drawing. These are referred to as neuraminidase, because we have certain drugs which we call as neuraminidase inhibitors. So remember this properly. So these are referred to as neuraminidase. Right. Now, one more thing that I would like to point to you over here is that these viruses, okay, let me draw first the capsid, and then we have got the genetic core. All right. So one more thing that I would like to point out over here is that apart from having all these structures, this particular virus also has got M2 proteins. Now these M2 proteins are nothing but your ion channels. All right. And these ion channels, they are present like this over here. And do you know what they do? They cause influx of uh, an electron, or sorry, not an electron, a proton, and it causes the influx of H plus ion inside. So through these M2 proteins or M2 ion channels, H plus ions come inside. Now, why do they come inside? What is the importance of these H plus ions? Let us look at in the cycle. So in a similar way, let us draw a host cell first, a big host cell with a dent and then with an involution representing endocytosis. So this particular virus particle, it selects its location on the host cell and it goes and sits over here, right? So it has got its hemagglutinin, right? And also it has got its neuraminidase. And then if you want to, you can draw your capsid layer and then you can draw your core genome. Now, uh, in viruses other than the influenza virus, what happens is this genetic core, right? It enters the cytoplasm, right? And it starts its own replication and translation process within the cytoplasm. But in the influenza type of virus, what happens is this genetic core has to be transported into the area of the nucleus, right? So that is what we are going to do. So first, what we are going to do is we are going to take this particle inside, right? And when we take this particle inside, we are going to draw the process again. And let me just draw the important things. And we have got your spikes, right? And we have got your neuraminidase. Now, here comes the most important part and what we discussed about the M2 proteins. Now, these M2 proteins, what they do is, now here, let me just write it, the genetic material. Now, these M2 proteins, these are your M2 proteins, right? Now, these M2 proteins, what they do is they cause influx of proton inside, right? They cause influx of proton inside. Now, this influx of proton is actually responsible for the process of uncoating, for the process of uncoating. Only when the process of uncoating occurs can this genetic material be released into the cytoplasm so that it can be taken into the host cell DNA area. So since now we have opened up the M2 proteins ion channels, the protons have come inside, and when they come inside, they will initiate the process of uncoating. So look at this, the capsid layer and the other layers are removed, and what remains is your core genome containing the genetic material. So this is called the process of uncoating. And how did it proceed? It proceeded with the help of M2 proteins that caused the influx of proton, which caused uncoating. Right. Now, this particular uncoating process will result in the genetic core being taken inside the host nuclear area. So what is happening over here? This is your viral strand of RNA, and this viral strand now is being acted upon by RNA polymerase. Now, this RNA polymerase is your viral RNA polymerase, and when it acts, it causes the production of your mRNA. Right. Now, the same process occurs when the mRNA is formed. The process of transcription translation proceeds with the help of the host cells ribosomes. Right. So this is your mRNA that has come, and now when when it is getting transcribed by the ribosomes, then you get two types of proteins, and those are your structural proteins and your non-structural proteins, non-S proteins, right? Now these structural proteins, these are important for the assembly of new virions, whereas the non-structural proteins, they are responsible for the production of more viral RNA. Right. Now, after this, the same procedure occurs wherein you get your regulatory proteins, then you get your structural proteins, and then you get your viral enzymes, and you prepare the virion. Then after this process, the virions are released. Release of virions. Now over here, there is one more step. There is one more way by which replication of viral DNA can take place, and that is through the replicative form of cRNA. There is one more area that you have to put out, and that is there is viral RNA polymerase acting again. Please switch off your audios. Now we have got another viral DNA polymerase that forms what I would like to call as complementary RNA. Right. This complementary RNA is again acted upon by RNA polymerase, and it forms viral RNA. Now this viral RNA helps in the production of structural proteins. So we have got a single strand of RNA or the genetic material that is acted upon by viral RNA polymerase. It forms mRNA or messenger RNA. It goes through the transcription process in the ribosomes and helps in the production of two types of enzyme proteins, structural proteins and non-structural proteins. Then here is where your assembly of virions takes place, and the end result that we have is your virion with the core genetic material, and then we get the release of virions. Now, another way of producing the viral RNA copies is through complementary RNA. So there is one way of producing viral copies is through mRNA, and the virus has got another backup plan, and that is through your complementary RNA. Now, this is initiated by the complementary RNA using viral RNA polymerase, and hence more copies of viral RNA are formed. So this is in particular the replicative cycle of RNA viruses. Here we have taken the example of influenza. So now let us talk about the pharmacology. The first set of drugs that we have over here are called as mRNA synthesis inhibitors. So let me write this as second, because the first was the DNA polymerase inhibitors. Now we have got mRNA synthesis inhibitors. Now as the name itself suggests, you might very well know where these drugs are going to act, and they are going to act at the level of the viral RNA polymerase. So we have got two drugs to remember over here. First is your ribavirin, and secondly we have got your formivirsin. Right. Now this ribavirin will act at the level of the viral RNA polymerase. So I'll write here as 2A, signifying ribavirin. Right. Now this formivirsin also acts at the level of RNA polymerase, but it has got a different action. What it does is it inhibits mRNA by binding to it and blocking it. Right. So it inhibits mRNA by binding. So here I'll write 2B. Right. Now, the third type of drugs that we have, over here are called as the drugs or inhibitors of viral penetration and uncoating. So now you very well know where these drugs will act. So before that, let us include the drugs. And the first set of drugs that we have are your amantadine. If you are thorough with your central nervous system, you will know where amantadine you have come across. And the other drug that we have is rimantadine. And this drug is quite unique. It is called Dr. Osanal. So it's Dr. Osanal. Now, since this particular group of drugs says that they are the inhibitors of viral penetration and coating, you very well know where they are going to act. They are going to act at the level of the M2 proteins over here. So they are going to block the M2 protein ion channels to open up. So if they don't open up, the H plus ions cannot come inside, and when they cannot come inside, the process of uncoating cannot start. And if the process of uncoating cannot start, this particular genetic code cannot be sent into the host cell DNA. Right. Then we have got the last type, and that I would like to call as Neuraminidase inhibitors. Now, Neuraminidase enzyme, what it basically does is Neuraminidase inhibitors. Neuraminidase, it helps in the release of the viral progeny. So when you are inhibiting this particular structure, all right, this particular structure is getting inhibited, you cannot expect this particular step to happen, the release of virions. Right. So we have certain drugs which are called as zanamivir or zanamivir. Then we have got oseltamivir. Then we have got the lastly peramivir. So this is about the different types of drugs and its classification with basis based on the antivirals of non-HIV type. So now let us talk about the individual drugs over here. So the first set of drugs based on the classification that we had were the DNA polymerase inhibitors, and that were your purine analogs, right? Now the first drug that we have to come across, and that is which is very important over here, is the acyclovir. Now this acyclovir, as you have already seen in the previous slides, so let us talk about it in detail. It is basically a guanosine analog, right? It is a guanosine analog which is converted into its active metabolite via 3 phosphorylation steps. That means when you take acyclovir, it is bombarded with phosphate molecules. Phosphorylation of these acyclovir is done. So what happened over here is that first up, when the cell takes up this particular drug, there's a enzyme of a virus which is called as thymidine kinase. Kinases, as you know, they are full of energy, they provide energy, and it converts this acyclovir into acyclovir monophosphate, right? And then the host cell possesses another enzyme which is also a kinase, and it is called as guanidulate kinase. Now this guanidulate kinase converts this monophosphate into diphosphate and finally into a triphosphate. Now this particular triphosphate is the active metabolite. And as we have already discussed in the previous slide, this active metabolite, it acts by inhibiting the viral DNA polymerase. So this is how acyclovir acts. Other drugs of this family, like your acyclovir, valacyclovir, and you have got ganciclovir, they also possess the same mechanism of action. Now let us talk about the pharmacokinetics of this particular drug, which is your acyclovir. Now this acyclovir has got, it is orally administered, and it basically has got 20 to 30% of its bioavailability. Now, apart from this, the plasma half-life of this particular drug is 3 to 4 hours, and about 90% of the dose is excreted unchanged in urine. Now, after hearing about its this particular pharmacokinetics, you have to make sure that you know certain specific aspects of this drug. Now, apart from having this particular points about the pharmacokinetics, you have to know that that I mentioned that it has got a plasma half-life of 3 to 4 hours, but in cases of renal failure, the plasma half-life goes to about 20 hours. So we have to make sure that when you administer patients or acyclovir to patients with renal failure, you have to be very careful. So, and one more thing, since it has got 20 to 30% of bioavailability, the peak plasma concentrations of this particular drug are sufficient only for the prophylaxis or the treatment of highly susceptible infections, such as your genital herpes, right? So remember these points about genital herpes and you giving acyclovir in patients of renal failure, because the plasma half-life extends to about 20 hours in such cases. Now, the antiviral spectrum is very much big, and it includes the very big family of the viruses, which is your herpes virus. Now, this acyclovir is highly effective against herpes simplex virus 1, which causes herpes labialis, or which you can also call as cold sores. And it causes herpes esophagitis, herpes keratitis, and also it can cause herpes encephalitis. So you can use acyclovir in the infections of herpes simplex 1. Then you can also use this in infections of herpes simplex virus type 2, which causes genital herpes. And then, as you all know, varicella zoster virus, which produces chickenpox and shingles. It is comparatively weak against the Epstein-Barr virus and the cytomegalovirus. Now, if you want to remember, the oral dose of acyclovir is 400 milligram TDS, right? So 400 milligrams 3 times a day is the usual dose. Now, coming to the adverse effects, there is no particular serious toxicity seen with the oral dose and topical use of acyclovir, but when you give it IV, it can cause phlebitis. It can cause rashes and it can produce a mild hypotension. Now, one thing that you have to remember is that cyclosporin, and the cyclosporin will be coming across in the next chapters, and other nephrotoxic agents should not be given along with acyclovir, because they may increase the risk of renal toxicity of acyclovir. And one important two marker that we have over here, which will be coming time and again, is about probenecid. Probenecid inhibits the renal excretion of acyclovir. So the question will be, the two markers that can be asked as a general one will be, name some drugs which probenecid inhibits. So you have to mention the list of drugs. So first list that you have got over here is your acyclovir. Probenecid inhibits the renal excretion of acyclovir.